Hello, I would like to warmly welcome everyone that is attending this week's edition of Extracellular Vesicle Club. EV Club was established by Ken, but is now an event of the International Society for Extracellular Vesicles. My name is Metka Lenassi, and I'll be your host for today. And it is my great pleasure to welcome today's speaker, Mattia Morandi from Ori Avinoam and Nita Regev-Rudzki groups at Weizmann Institute of Science. The focus of Mattia's talk will be on the use of cryo-electron microscopy to visualize EV fusion. And I think this is definitely a very important topic in the EV field, and that's why I'm looking forward to learn more about that. I encourage everyone to place any comments or questions in the chat box during the talk, and it will be possible to unmute yourselves at the end and ask some of those questions in person. And now over to you, Mattia. Please share your screen.
There you go. Thank you very much, Metka, for the introduction. First of all, I want to thank the EV Club, Kenneth, you, and everyone at ISEV for giving me this great opportunity to present this work, which we published in bioRxiv a few months ago, is now currently under revision, so hopefully will be out in the journal soon. And as Metka pointed out, the EV fusion indeed is quite an interesting and a very exciting field to study. And we've been working on it for the last few months. The way we got there and why it's important to study EV fusion, I think, arises from, first and foremost, if we think about the role of extracellular vesicle. I'm not going to give a very lengthy introduction to EVs given the context. Just we know that they're essential for cell-to-cell communication, and among the many reasons why I found it fascinating is that they are a conserved process that cells utilize to share information and transfer cargo, and you will find it across system and species. The fact that in pathologies, their role changes, their signature cargo changes to promote the onset of the pathology, but also for their potential role in therapeutics and diagnostic. And all of these aspects, if you think about all of these aspects, the cargo delivery process is really essential. From the fundamental EV biology point of view, obviously, the fact that they are cell-to-cell communicators and the fact that they can transfer content between cells relies on the fact that they can do cargo delivery. But even beyond that, if we think about the aspects that I mentioned before, the concept of cargo delivery already poses very interesting question or aspects. So, for example, we think about the fact that EVs are conserved across species and system, make us think, is the cargo delivery process itself also evolutionarily conserved, or does it change? Or even does it change or drastically modifies on the onset of pathology or in several diseases? And finally, once we know what this cargo delivery process is, can we harness it to improve the therapeutics aspect? You use EV as drug delivery vehicles, for example. And so this is just to mention that understanding cargo delivery is essential for our understanding of the biology. But if I were to present our current knowledge of today on the cargo delivery process of EVs, it would be a bit unbalanced, in the sense that we know a lot about the uptake process, and the general consensus so far is that the uptake of extracellular vesicle occurs primarily via endocytosis. But we don't know much about what happens post-uptake. So the pivotal moment where once the EV enters the cells in the endosome compartment, then the cargo is delivered. One thing for sure is that the fact that the EVs reach the endosomal compartment implies that for the cargo to be transferred intact, it must escape or it must be released prior to reaching the lysosome, because otherwise it would be degraded. So somewhere during endosomal maturation, between early to late, the cargo must be released. Something must happen that allows for the content to be transferred into the cell cytosol. And the way people have looked at it is by comparison, by simply observing that a similar fate post-uptake occurs for envelope viruses. And indeed, if we take a step even back further, we can observe that the parallelisms between EVs and viruses are not limited only in their post-uptake scenario, but really encompass the full life cycle from biogenesis, where we would have EVs and viruses that are assembled or produced either in organelles or at the plasma membrane, to their morphology, and finally, their delivery process. And the difference is that for viruses, we do know what is the cargo delivery mechanism, and that is membrane fusion. Particularly for envelope viruses that deliver the cargo at the endosome, what occurs is once the virion is internalized through the endocytosis, then reaches the late endosome. Now, endosomal maturation goes alongside with a drop in the endosomal luminal pH to acidic values. This acidic pH triggers viral glycoproteins on the surface of the viral envelope, which in turn mediate membrane fusion between the virion membrane and the endosomal membrane, allowing the cargo to be transferred. So given all the analogies that we can observe and think about between EVs and viruses, and the fact that we know that membrane fusion is the viral cargo delivery process, could it be that EVs deliver their cargo via membrane fusion? And indeed, the more recent literature does point to this hypothesis. In cellular, we have two primary works that show that membrane mixing of labeled EVs occurs at the endosome, and then the process is really dependent on a late endosomal lipid called LBPA. And similarly, Joshi and others in ACS Nano a few years ago showed that the cargo delivery also occurs on the endosome, and that is modulated by the pH. And most recently and interestingly, the group of Gregory Lavio in two separate papers showed that using an in vitro system, the release of the cargo from EVs interacting with plasma membrane sheet is pH dependent and it's protein dependent. And all of this does point to a membrane fusion that is triggered by the late endosomal environment. But the question remains that whether it's really fusion to an extent, like is it definitely proof? And definitely what we don't know yet is what is the machinery, what are the molecular components involved in this process. So in our work in the groups of Neta and Ori, what we decided to do is to focus on the first aspect, to try to dissect the fusion part. And the way we looked at it is by approaching it as people in the virology field approached the viral membrane fusion subject in the old days, and that using a completely in vitro minimal model system on viruses interacted with liposome, or LUV for short, and just by replacing the viruses with the EVs. And there are a few advantages and a few reasons why we want to do that. The first one is that the liposomes, which would be our target membrane of choice, in a sense, the target for the EVs to fuse to, are fully tunable. They are completely artificial, so we have full control about their composition, size, cargo, and as well, we have full control about the environmental parameters like temperature and pH and so on. And so we can really focus on the EVs as the only active and biological component in the system. So what we do is we isolate EVs from cell culture supernatant, and because we need really clean and pure sample, we go through 3 steps. First round of ultracentrifugation, then a density gradient, and finally sucrose cushion of the EV positive density gradient fraction to fully clean completely, and we can confirm it via nanosite, Western blot, and obviously cryo-EM, where we can confirm the morphology and size. And then we perform what is called a membrane mixing assay, which is based on FRET. So to give you a run through of how the experimental setup works, we label EVs with a pair of membrane dyes that are called a FRET pair, the I and the ID. And then we incubate this FRET labeled EVs with liposome that mimic the late endosomal membrane composition, containing primarily LBPA, a negatively charged lipid found in late endosome. And then we mix them together. If there is no interaction, no fusion, if we monitor the donor, the II, we should not see any change in the fluorescent intensity. The moment fusion occurs for any reason, in trigger or something else, then the fact that we are adding a non-label membrane to the EV membrane means we are diluting the dyes, so the fret efficiency drops, and therefore the donor intensity increases. And then we normalize the signal from the signal due to the fusion with addition of detergent, and this is considered infinity dilution, and this will be our 100%. So this is a typical kinetic profile of a membrane mixing assay, which is a proxy for fusion, and which has been extensively used as well instead of viral fusion. So if now we compare using this assay viruses and EVs, what we will see is that the profile looks very similar. There is no interaction prior to trigger. In physiological pH, minimal interaction for both of them, and this is the normalization with the detergent. But the moment we acidify to late endosomal pH, we see that for both EVs and viruses, a drastic increase in the donor intensity, signifying membrane mixing. And if we compare all the values for multiple experiments, we see that the fusion efficiency between EVs and viruses is comparable, and in particular is triggered by the low pH. Whereas if we check only liposome interacting between themselves, we see that there is no fusion, no membrane mixing at any pH. And this indicates that it's not just a spontaneous process between lipid bilayers, but it's something more. We know for viruses, the triggering of the viral glycoproteins. For EVs, something is triggered that allows the fusion. And we can confirm it in another way, complementarily, using nanocytes. So if we, for example, measure the size distribution of EV liposome mixture at physiological pH, and upon acidification of the environment, we see the appearance of a higher diameter population. And overall, if we quantify the average diameter, we see that liposome at any pH are very monodisperse, but EV liposome mixture upon acidification increase their typical size. So this implies that multiple vesicle are adding onto each other by fusion, and therefore result in a larger diameter vesicle. And this is really consistent, this acidification process, with what has been previously observed, that low pH is required for cardio delivery, and in this case for fusion. And we show furthermore that this process is protein mediated, because if we treat EVs with proteinase K prior to incubation with liposome, we see up to 70% reduction in the membrane mixing. So the proteins are required for the fusion, and we can confirm this is not due to a destabilization of the EVs, because first of all, we shave only the membrane proteins and not the luminal proteins, but also the size distribution does not change so drastically. So EVs are still intact, but their ability to fuse is significantly limited. So all of this well recapitulates what has been observed before in the literature in terms for cargo delivery and in vitro for fusion or let's say content release. But now we can take it another layer further, because we can take advantage of the fact that we have two vesicular population, and we can study the interaction between EV and liposome using cryo-EM. So to give you an idea, this is our typical grid for cryo-EM looks like, and we can really do a semi-automated workflow where we can acquire the whole grid or the individual squares at medium map, and this will allow us to identify points of interest, which can then be acquired of higher magnification and high resolution, where we can distinguish the bilayer. And now a question that I get asked often at this point is that how can you distinguish EV from liposomes? Serendipitously, in our analysis, what we found is that you can actually tell them apart. So if we observe, for example, this is what we think it's the EV liposome interaction. And what you can tell is that this gray distribution of the luminal part of the vesicle is much smoother than this one. So this one is more of a granular spatial distribution of the gray intensity, and this is because we think liposomes don't contain anything inside because they're artificial, so they have only buffer, whereas EVs have a biomolecular cargo, and this gives rise to a specific contrast due to the cryo-EM. But obviously this is qualitative, but if we do a proper analysis by doing a radio distribution profile of the gray intensity values between EV only or liposome only, we can see really that you can distinguish them. So liposome, because they have nothing, they're empty, their spatial distribution is much smoother, it's more like stochastic noise. But for EVs, there is a periodicity that arises from this granularity in the luminal signal. And really, this way we can look primarily at only EV liposome pair and look at what kind of interaction occur. So the first one we observe is a very tight adhesion where the bilayers are in tight junction, but still separate. There's no fusion yet. You can tell because if you run here, you will say the inner leaflet of the EV, the inner leaflets of EV and LUV, and finally the outer leaflet, sorry, of EV and LUV, and the inner leaflet of the LUVs. So there are two separate bilayer in tight contact. But then also we start observing proper fusion intermediates. So for example, hemifusion, where only the outer leaflets are merging. So if we look here, you will see the bilayer of these EVs and the bilayer of this liposome, and you should look only at the outer bilayer. And you can see that they merge together into an individual bilayer. So this is what we define as an hemifusion. And this is just mixing of the membranes. And then we see finally the appearance of the fusion pores as well. And this allows for the lumens of the two vesicles to be in contact. Now this will be proper cargo delivery. So when the content can mix with each other. And this finally results in what we call a post-fusion or an expanded pore intermediate, where now basically it will be difficult to distinguish the two vesicles from each other with just one continuum. And so this is a bit of the array of all the possible interaction that we have. And this is very well in line with is being reported for viral fusion. And if we compare, for example, to liposome only interaction, we see that no fusion intermediate occur in liposome at any pH. And even when adhesion is present, let's say contact between the vesicles, it's not at the same extended contact interface that we've observed for EV liposome interaction. And now, because we can have a semi-automatic workflow where we can acquire multiple position and multiple EV LUV pair, we can do some statistic in it. And really, we can see that, obviously, as I show you for liposome, really nothing happens and nothing changes whether you are at just pH 7 or pH 5. There is no fusion. But more importantly, that when you look at the EV-LUV interaction, the real fusion intermediate, so where there is merging of membranes and mixing of the lumens, occur only at pH 5. So structurally, we see that it's fusion, and most importantly, it's confirmed that this is a fusion that it's pH dependent, and because the assay also works for the membrane mixing, we know that it's also protein dependent. And now this system it's already prepared for cryo-EM. That means that we can actually do cryo-electron tomography, meaning we can tilt the sample at different angles and acquire it and then reconstruct it to get a 3D volume. And in this way, we can remove any ambiguity for the fact that we are looking at 2D images of a 3D sample. So to give you a few examples of what we see, this is, now we are looking at the animation. It looks like a video, but actually we are slicing in Z through the 3D space of the vesicle liposome interaction. And what you can see, as soon as we appear again, is that we actually see, we were lucky in seeing two EVs interacting with the same liposome. So we'll see this one, and you can see the merging of the membranes together, but also this bottom one is, you can see a connection in the bilayers between the liposome and the EVs. And this is sort of like a still frame of one of the slides to give you an idea. And there is an additional one that is sort of similar, but really reinforces the point that there is a fusion pore open connecting the two lumens. So this is really fusion, and this, of course, happens only at acidic pH. And to reiterate the point, these features are very, very closely similar to what has been reported for viral liposome interaction. Now, obviously, viruses, as you can observe here, are much more easy to distinguish and detect than EVs in a cryo-EM sample due to their shape, but also their very thick protein corona. But if we look at the interaction and in the fusion intermediates, there is a very close analogy. And this also was done for a virus that fuses at low pH. This was influenza virus image from the group of Kelly Lee, University of Washington. So really the EV fusion is, from an intermediate point of view, indistinguishable almost from viral fusion. And now that we have this confirmation that our assay with EV liposome really recapitulates the pH dependency observed for cargo delivery, and the protein dependency observed for cargo delivery, but most importantly, it tells us that it is fusion with one additional step, which is that the fusion is unilateral, meaning much like viruses, EVs do not require necessarily a component on the other side beside a membrane to be able to fuse. We can start playing with the system and asking some question about what modulates it. So one aspect that I mentioned before in the introduction was the lipid composition and the fact that the late endosomal LBPA was required for the cargo delivery in cellular. And this is quite important because endosome maturation does not only imply change of pH, but also implies a very significant change in lipid composition. So from early endosome, where you would have a lipid composition more closely similar to an outer leaflet of the plasma membrane, so reaching sphingomyelin, sphingolipids, and cholesterol, as it matures, the endosome gets enriched in LBPA, but gets depleted in cholesterol. So one question we can ask is, how does lipid composition affect the fusion, knowing that now with our assay, we are not probing cargo delivery in general, but we're probing fusion efficiency exclusively. And what we see is that the further away we go from a late endosomal lipid composition, the lower the fusion efficiency gets. So if we go from early endosome with sphingomyelin cholesterol, it's still lower than a late endosome lipid composition. And if we go to a fully non-biological membrane composed only of DOPC, the fusion efficiency is really, really low. And even more importantly, we can tell that the affinity for a late endosomal lipid composition is not due exclusively to the fact that we are working with a negatively charged lipid. So it's not an electrostatic interaction-based fusion or EV liposome interaction, because if we swap LBPA for phosphatidylserine, which is similarly negatively charged, but obviously is not present the same amount in late endosome, the fusion drops significantly. So there is an affinity specifically for LBPA and for late endosome. And finally, we can address one question / paradox in the concept of having a pH-dependent EV fusion. And for this, I should thank actually Guillaume Vanier, because he's the one that first posed me this paradox when we met at the EMBO course a few years ago, which is, if EVs fuse triggered by low pH, how is it possible that they are formed in an acidic environment? And this is a good point, because part of the EVs are formed in the multivesicular body, which is also acidic. So I didn't go over it in the part of viral fusion, but what we know from decades in virology is that acidification of viruses without a membrane to fuse to results in their complete inactivation, because it's an irreversible process. So this is a paradox, because how is it possible that EVs are formed in an environment that at best will inactivate them, or at worst will wreak havoc in the multivesicular body? So we didn't have a specific answer, but what we decided to do is to take an unbiased approach. Basically, we took EVs, we pre-acidified them on EVs for a certain amount of time, and then we reneutralized back the pH to 7.4, and then we performed the incubation with liposome and the fusion assay. And what we observed was very interesting, because obviously, and of course, we compared that to viruses. if you take envelope retroviruses, which work at low pH, and you inactivate them, then you pre-acidify them, their ability to fuse is drastically inhibited. EVs, on the other hand, do not get inactivated. Their ability to fuse is retained even upon pre-acidification. And this is quite interesting because in nature, there are a few viruses actually that are able to perform such a feat. The most prominent one is vesicular stomatitis virus, VSV, which we use as an additional control. And the fact is that VSV has a very specific structure of its viral fusion protein. So we think this might be a hint to what the structure of the potential EV fusion protein could be. And of course, this would be very interesting if proven, because it solves a lot of paradoxes along the EV biogenesis way. So with this, I will conclude. And just to summarize that EV indeed are capable of fusion to artificial membrane in a process that it's unilateral. EVs are capable of doing just by themselves, pH dependent and protein mediated. And this is highly reminiscent of viral fusion of many envelope viruses. This process is compatible with biogenesis in the MVB or in acidic compartments and may hint at what the structure of the viral EV fusion protein could be. And additionally, we showed that what has been shown for the membrane lipid composition affecting cargo delivery, now I can say quite confidently that actually that affects, much like viral fusion, the EV fusion. This is a feature that has been observed very extensively for viruses, where the target membrane lipid composition really affects how efficient the fusion is. And this obviously opens a lot of new avenues, or opens a lot of doors to, now that we have a system that focuses only on measuring fusion, we can utilize it to screen and let's say test different protein candidates to try and identify the EV fusion protein machinery. And on top of that, I think what is also very interesting, which connects to my introduction at the beginning, is this is work so far has been done on human cell lines. And it will be very interesting to see if the fusion mechanism and the fusion triggers and so on are different in different organisms. And also particularly even within the human cell lines, we know that different EV subsets exist. Big question that I found really intriguing is how these different subsets could be different from a fusion standpoint. And to give you just a snippet of a partial answer to these two last points, we published this month in the work on malaria-derived EVs, and we performed a similar fusion assay, and which were led by Paula, a very talented PhD student in the lab of Neta. And what we saw is that the triggers and the preference towards membrane lipid composition is different than in human cell line. And more importantly, because we were looking at two different EV subset based on size, fractionated by size using asymmetric feed flow, we saw that also the fusion efficiency at different condition is different between subsets. So really, I think this opens many avenues in trying to understand how EVs may deliver the cargo in different conditions, different organism, and perhaps also in different pathology. This is quite an interesting aspect as well. And with that, I would like to thank my two labs, the lab of Neta, the lab of Ori, for all the support and allowing me to perform this work at the intersection between, say, biophysics, EV, and membrane fusion. Petro, who helped me very much with the project, and everyone else was involved, in particular the group of professors Gil from Technion, who contributed a lot in terms of their expertise, and all of you for your attention. I would be happy to answer any question.
So thank you, Mattia, for this really very, very interesting and I think a talk. And there's even some applause and a lot of questions.
Thank you.
So yeah, so really, and also beautiful presentation. So that was also we saw that. So maybe first Philip can start with his questions. I think that you can unmute yourself.
Yeah, it's muted.
Okay.
It's a very fine and careful presentation. For me, there is a duality. There are subpopulations of EVs that can pass the stomach acid enzyme environment and in our hands end up having actions in the skin. And there's other instances, of course. So I wonder about how that compares to what you think of the liposome. And I'm troubled that the artificial liposomes can't really imitate the real thing, the liposome itself. It's artificial.
It is artificial. And of course, even if we look at the field of virology, this kind of assay or this in vitro system do not recapitulate all of the fusion and cargo delivery process. So for example, most viruses or a lot of viruses would require cleavage or a proteolytic site to activate, to be primed for fusion, so to speak. Still, even without cleavage, you will see some degree of fusion to liposome. So obviously we are looking only at the partial component of the V fusion. It's not 100% what happens in vivo in the endosome. But I think from a structural point of view, it tells us that it's fusion, which is already a big thing, because this kind of assay, I mean, if there is a trigger and it occurs in fusion, fusion structurally does not change between systems. It's a very mechanistically conserved process. So we can probably, the surrounding aspect in terms of what triggers it, how it's regulated, it's probably much more complex in vivo, but the process itself of fusion, I think, is retained both in vivo as it is in our in vitro assay.
I'm not questioning the fusion you have shown, but it seems to me there are systems or subpopulations of EVs that resist the acid enzymes in the stomach and probably may resist the acid enzymes in real liposomes
sorry. I saw, you mean the lysosome? Yeah, it could be. I mean,
the liposome is a totally artificial thing. The lysosome has many, many components.
Yeah.
And there are some populations of EVs that resist acid and enzymes.
So yeah, this is a very good point. I mean, that's why I think it's very interesting to look at EV subset specifically, because as I mentioned, we are looking at the bulk. And if you notice in our Western blot, we had both CD9 and CD63. We cannot tell which is which in driving the fusion. But you're right on the point. There are probably different routes, and different EV subset might have different affinities, and one might not fuse, or might just be going to the lysosome and deliver their cargo through
some other means.
Yes, and actually this is still a big question in the field, because obviously from my standpoint, I look at fusion and knowledge to viruses, but there are also arguments towards a transfer, like a passive transfer or a core or some kind of protein-mediated transfer that is not fusion-based.
Another potential weakness is using the EVs from cell lines compared to real EVs from primary cells.
So we are trying with plasma patient sample, and to an extent we see also fusion there. Problem there is that it's a mixture of many sources.
Yes.
So once again, I mean, we can see that there is fusion, but obviously we'll not be able to pinpoint which part of the mix of EVs will drive the fusion mostly. Or if
the subpopulations is a major challenge.
Yeah.
So I think that
Thank you very much.
I think that
Thank
you
you.
just identified your project for the next few years, right? So a lot of work still to be done. And I'll just read a question from Ursula because she doesn't have a microphone. So she's asking, in your preparations, do you also observe EV-EV interactions and fusion events? And
Yes.
if I may add something, because it's very similar to this, like in urine, the pH can also get very low. So would you expect in urine for EVs to also fuse if you don't neutralize the pH while isolating it?
So that's a good question. So the answer is yes, we do see EV-EV interaction. We excluded, obviously, we do not see in the FRET assay because all the EVs are labeled, and because the FRET is based on the dilution of the dye, EV-EV interaction is not detected in the assay, but we do see it in the cryo-EM, which is actually good because it tells us that even in members that are more decorated, so to speak, that have membrane protein and so forth, the fusion is still possible. So it's quite good. Now, regarding to the pH, it depends on the pH. To be honest, I don't know what's the pH of urine.
Oh, well, I think that it could reach 5 or something, it depends.
Yeah, so
there can be quite
depends.
A range.
Yeah, so it depends on a few factors, to be honest, because so one of the things that I didn't show here is that
urine pH is 7. Around, it's neutral.
Sorry.
So somebody mentioned that the pH of urine is 7. It's neutral.
Well, okay. I don't know. At least when we're working with that,
Not all the time.
I can help. I'm a nephrologist.
Okay, great.
Please.
Perfect.
One of the important things of the kidney is to get rid of protons, to get rid of acid. So urine pH on an average is between 5 and 6. It can be even drop below 5 under special circumstances.
Okay.
Okay. Thank you for supporting what we see in the lab.
I wouldn't be able to. So, but I can tell you that, so this assay, obviously, being in vitro, what we do to sort of increase the output is we pre-incubate the EVs with the liposome at 4 degrees for half an hour, because it allows to maximize the contact between them. So that tells you already that the fusion event it implies that the EV might be in contact with each other. So it depends on the concentration, really, and the time in which you maintain them. But a highly concentrated system will determine how many fusion events you have. If you have an incredibly diluted sample with very low concentration in your vesicle, even if you acidify, the chances that they come into contact to each other in the time frame that the fusion can happen, it's still limited, I would say.
Okay, perfect. And we have Rihanna. She has a question, so if you will unmute yourself, please. Rihanna, okay, maybe then I'll just read it. She was asking if you check the stability of EVs and liposomes at pH 5 separately.
Yes, we did, and they are pretty much the same. Obviously, with the EVs, we do see sometimes some fusion events when you just have EV alone and incubate, but we don't pre-incubate them together at 4 degrees, for example. So they are pretty stable in the sense that they don't get, especially the liposome, they don't get damaged, they don't get deformed or change their morphology drastically.
Okay, so then Mikhail has a question. Mikhail's clear?
Yeah.
Okay, perfect.
Yes, hi. So I'm curious about the effect of surface decoration, like membrane proteins, on the whole process. When you look at liposomes, if they have brush on them, that usually prevents them from fusion, so that has sort of steric stabilization effect. And of course, in all your pictures, you don't really see membrane proteins, and I'm not sure if your artificial vesicles even have those. So I'm kind of curious about your general comment about the sort of effect of surface decoration on a fusion. And the other thing that I sort of noticed when I was looking at your results is that after you shaved your EVs by treating them, by digesting them with protease K, your results don't show a reduction in the hydrodynamic size, which, in our experience, and other people reported that as well, after that treatment, the hydrodynamic size reduces quite substantially. Thank you.
Okay, so to respect to the first part, actually, well, in the images I show you can't really tell, but in some cryotomography you do see protein decoration on the surface of the EVs. It's much sparser than you would see in a virus, because they're just showing some images. The virus is very dense brush, but you can still see some protein on the EV surface.
Probably like shadows, right? Like sort of excess density kind of showing.
Yeah,
up.
it's just, they're not so structured, but you kind of see some dots surrounding, especially in tomography, because you're reconstructing, you can see it a bit better. In respect, and obviously the artificial vesicle have none, the artificial vesicle have only lipids. With respect to the treatment, I would not be able to give you a proper answer, in the sense that we measure it, and the drastic was not a drastic change in size. I mean, I show you representative. I haven't looked at the average size distribution, but I would be a bit. I mean, when you say drastic, what do you mean? Like,
well, it could be 20, maybe even 30%.
Okay.
It actually depends. Actually,
percent.
pH. So yeah, so pH actually would impact the conformation of your membrane proteins. So in fact, you could even things like osmolarity would affect. You would see through the change in the sizes, right? And certainly pH affects that too. So in fact, proteins may be even changing there. So this is just something I was thinking as you were presenting. And the other thing, and we may not be able to resolve, the other thing that was kind of curious to me, at this sort of dimensions, curvatures often matter.
Absolutely
so it
right.
may be very different when you look at diffusion of the EV with something that looks flat, like, for example, plasma membrane. Right.
Yeah.
And even though you call them large, I forget what you call them, those lobes. They're actually quite small. Curvatures are actually quite
so
steep.
You are absolutely right. So I would just mention that it's called large in the field of liposome because large is 100 nanometer, and you would call them small at 50. And if they're larger, they call them giant.
Yeah.
So it's just a terminology thing. You're absolutely right in the sense that there is a curvature effect. However, it actually probably the opposite way than you might think, because high curvature systems are not necessarily easier to fuse to. Flat membranes might be easier. So, and for viruses, we know that if you go from 100 to flat membranes, the difference is not so huge. But definitely what we are looking next is to liposome with different size to see if there are difference in the fusion efficiency of EVs. If you go from 100 to 50 to 400, for example, or more, how drastically the fusion efficiency could change.
Okay, perfect. And Winfried also had a question.
Yeah, hello. Sorry, I have to check if my camera also
we can
works.
hear you.
Yeah, it's fine. It's more a very general question about can you, based on your results, can you describe your thoughts about in how far fusion can really also occur on cellular plasma membrane and EV interactions, or is it really just that we have to assume that it's only endocytosis, and in how far could be this also related to the idea that maybe EVs can also transcytose, because this is also a big question still to be answered, I guess.
Yeah, it's a good point. It's a question that also viruses, virology field has to face. I don't exclude plasma membrane fusion for a few reasons. First and foremost, the fusion process, it's triggered, but the trigger itself drives the kinetics of the process, but it can also happen over long term, not necessarily a low pH. So fusion at the plasma membrane is still possible. Matter of fact, I think people have shown that, I mean, the pH just increases the chances of happening. It just so happens that people have shown in, I want to say, tumor niche cells, that the fusion of the plasma membrane is higher. And this is due to the fact that the extracellular microenvironment is slightly acidic. So that could help, but definitely plasma membrane fusion occurs. We show it for the malaria liposome, for example, that plasma membrane is the best composition rather than endosomal. So my feeling towards it is that it could really depend first and foremost of the source. In malaria, we sort of rationalize by the fact that it has to target a non-endocytic cell, the red blood cells. So the fact that it's preferential to plasma membrane does make sense. If different cell types could have preferential compositions, it's also possible, much like some viruses do. HIV that fuses at, like for example, HIV, it's a bit different system, but HIV could fuse at the plasma membrane, but part of it can also fuse at the endosome.
Hello, and very interesting talk. I really appreciated it. The experiment that you did where you pre-acidified and then reneutralized and then did your fusion, how long was it? How long was the period when it went back to pH 7? And have you ever tried varying that? Basically, my question is, how long does that fusible state last after you've returned to normal?
So the system is 1 hour each. So 1 hour at pH 5, 1 hour at reneutralize to fully assume complete reversal of the pH. I didn't try changing it, I'll be honest. So I don't have an answer specifically to what I could expect when this state lasts. For viruses, I know it's irreversible. In the sense, once you do it, there's no way. For EVs, not sure.
Because knowing that might give you an indication of what its mechanism is. If it's temporary, it only lasts 1 hour, 2 hours. If it's irreversible, then you're looking at something in a protein structure likely has irreversibly changed.
Yeah, so for example, for VSV, it's a bit trickier than that, because for VSV, it's protein conformational change. It just so happens that the protein conformational change is reversible. So if you imagine viral glycoprotein are sort of like elbows or hinges, so they extend and then they revert back. And this reversion is what is irreversible. So the fusion, the pH sort of triggers the opening of the hinges. The reversion is spontaneous and it's irreversible, but just so happened the VSVG has a metastable state. So if I remember correctly, it opens, it returns, but then it returns primed that it can open again. And so, I don't know if it's timed, and we never tried to change the times to see if different times could make them completely irreversible or not. Actually, that could be an interesting thing to try.
Also, have you tried looking at a gradient of pHs, steps 5.5, 5.2, 5.6?
Yes. Yeah, so we try the full array from 7.4 up to 5 in every 0.5 increment, and we see really a curve that slopes down at 5.5 and 5.
Okay.
So late endosoma, but yeah, we probe the whole range for all the viruses and the liposomes.
Okay, thank you.
Thank you.
Okay, that's perfect. And Ami has another question.
Yes, thank you for your presentation. It's an impressive presentation. My question is regarding to your cryo-EM images, and I want to know more about the resolution and the size of your images, and also the methods that you use actually to reduce the noise, because normally we don't have these shapes from cryo-EM, and you'll compare in one part the intensity of the pixels with the other thing. So I'm wondering that what the method you use to actually to obtain this kind of images. Yeah,
sure. So the 2D images are acquired at 200 kilovolt in Antarctica with a one view camera. Usually the resolution I use for acquisition is 0.4 nanometer. And with general dose of, there's not much, those you cannot really measure in one view, but let's say exposure time of roughly 1 second. Then to present the images in that are much clearer, what we do is for the full image, you can do a bandpass filter. We do a bandpass filter to remove all the smaller information and the larger ones, so that really the bilayer gets really nicely uncontrasted. The tomography, on the other hand, are acquired in 300 kilovolt with 0.2 nanometer resolution, but then in reconstruction we bin them for. Sorry, I don't want to get too technical, but just these images, we don't perform a bandpass filter on that, but I do a non-anisotropic linear diffusion filter on Itomo. Now the number of iteration and everything depends, but this is how they get much cleaner. With respect to the spatial distribution of the cargo, so at ISEV, I had this conversation as well, because not everyone sees it. Maybe we get lucky because our sources do it. In the malaria EVs, I also see it. My guess is that it depends a lot on the source as well.
Yeah, you're right. Thank you so much.
Okay, perfect. I'll just read the short question because I think we've already discussed it a bit. So have you, Leon asked if you've looked at microvesicles?
No, we looked only at small EVs.
Okay, perfect. And then Ursula has another question, and I'll read it because I remember she doesn't have a microphone. So does EV lipid composition affect the fusion? For example, do EVs enriched in ceramide or sphingomyelins have different fusion efficiencies?
That's a tricky one. I would not know. I mean, this is hard to speculate on, because it's, on one hand, it could be yes, because we know membrane proteins activity does depend sometimes in the lipid environment they're in. But this would be interesting to study, but I guess also a bit tricky, because we will have to shut down some of the synthetic pathway of the lipids. So I don't have an answer for that. My guess is yes, to an extent it would affect.
And then Hakai has the last question. So maybe if you can just... Okay, perfect.
Okay, great. It's a very interesting research. I hope this could simulate the cell, like a kind of, we call it in vivo situation, really your situation to isolate the lysosome itself. Another question is, you see these two vesicles bind together. So can you just label them to be sure that vesicle is the vesicle you're talking about, other than the lysosome, using some antibody gold and then combine your cryo-EV to confirm it, other than using the density
so
in this case?
In principle, you could. However, cryo-immunogold requires a few steps for cleaning that would affect, I think, a bit the quality of the sample, because you need to spin down, like to label properly a sample, you need to spin down. And so to do that in parallel to doing a fusion assay gets a bit complicated. What one could do that it's kind of easier is to do correlatives. So you could stain the EV in one color and the liposome in one color and look them in fluorescent, and then go in cryo-EM to look only at point where the two are. From my experience, we can actually tell them apart, especially in tomography. You can kind of, when you start seeing the protein corona, you can tell what is a liposome and what is an EV. And for terms of 2D, I think the cargo, the luminal spatial distribution, for us at least, was a very good indicator. I mean, it was really consistent. Okay. But obviously, there are more sophisticated methods where you can tell 100% what is what.
Sure, thank you.
Thank you.
Okay, perfect. Just a quick question from myself again.
Sure.
So I was wondering, when you change pH, you definitely affect so the charges on the surface, and these charges affect zeta potential, and zeta potential kind of like is supposedly, I mean, is a measure also how these colloidal particles will act in the solution. Yeah. So maybe if they will aggregate or interact or something like that. So do you see any changes in how these EVs behave if they have?
So I did some cryo-EM imaging of EV at different pHs, but I haven't looked at the aggregation, because we were looking at another thing. We were looking sort of the protein corona, hoping to find what you will see in viruses. Definitely for liposome, yes, because with liposome you see a bit more shallow aggregates, not as tight junction as the liposome, but still. But in terms of colloidal stability, no, I didn't notice drastic differences, at least drastic, let's say.
Mattia, I just want to thank you so much for taking your time and sharing your very interesting work on fusion. And I think that everybody enjoyed it, and there was a lot of discussion. And I hope that people will also join your talk on YouTube when it's available. And just, yes, good luck with your future work and your forming your own group, and just
finger crossed for that.
Yes. So thank you all for attending, and we'll see each other again in 1 week when Ken, I think, will monitor or another session. So bye.
Thank you very much. Bye.